Good morning children. In this video, we will going to discuss about testing for production of heat and carbon dioxide in anaerobic respiration. Children, we already discussed about aerobic respiration and anaerobic respiration. Anaerobic respiration means that respiration take place in absence of oxygen or low amount of oxygen. In anaerobic respiration, heat, carbon dioxide, energy, ethanol or lactic acid is released. For this activity, required materials are wash bottle, 2-hole rubber corks 2, thermometer, L-shaped glass tubes 2, mixture of yeast and glucose solution, bicarbonate solution or lime water, Jonas green bee or Daisy green solution. These are the required materials, children. Now we can look at the procedure of this activity, children. First we can take the wash bottle. That wash bottle, half of the wash bottle is filled with mixture of yeast and glucose solution. Yeast is a one type of fungus. It can perform anaerobic respiration in the absence or low amount of oxygen. This glucose solution contain some dissolved oxygen present in this mixture. That oxygen, we should remove the oxygen from the glucose solution. For that purpose, we can heat the glucose solution with a minute and then cool it without shaking. While doing this activity, one precaution is important to take. That is that don't shake the glucose solution after heating and then cooling. If we can shake the glucose solution, it may be chance the entry of oxygen into the mixture of glucose and yeast solution. So we should not shake the glucose solution after heating and cooling. Children, now we check the oxygen is present or not in the mixture of yeast and glucose solution. For this, we can pour the few drops of Daisy green or Jonas green bee solution in the mixture of yeast and glucose solution. This blue dye turns into pink when short supply of oxygen around it. This indicates this glucose and mixture of yeast and glucose solution does not contain oxygen. After that, we can pour the 1 centimeter layer of liquid paraffin. This liquid paraffin prevent the entry of oxygen into the mixture of glucose and yeast solution. After that, we can fix the thermometer into the one hole of the rubber cork into the glucose solution and fix the L-shaped glass tube in the another hole of the rubber cork. This L-shaped glass tube is attached to the another L-shaped glass tube. That L-shaped glass tube is inserted into the test tube. The test tube contains bicarbonate solution or lime water. We can observe the activity after few hours. Temperature raised in the thermometer. So we can conclude heat is evolved during anaerobic respiration. And another observation is also we observed, that is the bicarbonate solution turns into milky white. This bicarbonate solution turns into milky white. This indicates that evolved gas is the carbon dioxide. So through this activity, we can prove the heat and carbon dioxide is produced during anaerobic respiration, children. While doing this activity, we can take two precautions, children. The precautions are don't shake the glucose solution after heating then cooling. And another precaution, we should handle the thermometer carefully, children. So children, through this activity, we proved oxygen. In the absence of oxygen, anaerobic respiration can take place. In anaerobic respiration, heat and carbon dioxide is evolved, children.